Usually when we talk about a supercomputer, there's this expectation of cold, rigid precision.
Oh, absolutely. You expect sterile logic.
Right. You look at a modern server rack and you see the hyper-engineered silicon, the gold plating, the pristine copper wiring, and it all makes absolute logical sense. It looks like something explicitly designed to compute.
Yeah, it looks intentional. I mean, every pathway is optimized by an engineer for, maximum theoretical efficiency.
Exactly.
Yeah.
But then you step into the world of human neurodevelopment, and that entire paradigm of pristine engineering just gets completely turned on its head.
It really does.
Because we're looking at a biological landscape that is, honestly, it's an absolute mess. It's wet, it's fatty. It is basically the definition of organic chaos.
It's a remarkably noisy environment, and yet this wet, noisy biological system routinely outperforms those silicon server racks in ways that artificial intelligence researchers are still, desperately trying to replicate mathematically.
It's just wild. So welcome to this custom-tailored deep dive. We are so thrilled you're joining us today.
We really are.
Our mission for this deep dive is to demystify the ultimate supercomputer, which is the human brain. We have taken a massive stack of sources curated specifically for you.
And it's quite a stack.
Yeah, ranging from really dense lectures on functional neuroanatomy and evolutionary biology, all the way to some incredibly complex computational physics theories regarding something called active inference.
Right, and our objective here isn't just to, catalog what the brain is made of. We really want to understand the mechanics of how it processes reality.
Yes, and to set the stage for you, and just to give you a sense of the staggering, almost incomprehensible scale of what we're dealing with, think back to when you were in the womb.
This is one of my favorite statistics.
It's nuts. At peak development, your brain was growing at a rate of roughly 250,000 new nerve cells per minute.
Just let that sink in for a second.
Right, 250,000 cells every 60 seconds, just furiously organizing themselves into the structure that is currently allowing you to understand the English language.
And if we connect this to the bigger picture, it's not just about the staggering volume of cells. The core theme we need to keep coming back to today is that the brain is not a passive receiver of information.
Huh?
It is fundamentally an adaptation machine. It's an organ uniquely pressured by evolution to minimize surprise in a completely unpredictable, chaotic universe.
Okay, let's unpack this, because before we can understand how this machine predicts anything, we really need to look at what it is actually made of. We have to open the hood.
Definitely.
And looking at the biological hardware, it's immediately counterintuitive. Like, if you analyze the gross composition of the human brain, it is roughly 77% water. Another 11% are lipids, basically fat, and about 8% are proteins.
Yeah, it's essentially a highly organized saline solution suspended in fat.
Exactly. And it weighs anywhere from 2.6 to 3.1 pounds. To put that visually for you, that is roughly 3.5 to 4 times the volume of a regulation baseball.
Not very big at all.
No. So you have this watery, fatty mass the size of a few baseballs sitting in the dark, pressurized vault of your skull. And somehow, the fluid dynamics of that wet lipid mass hold your entire consciousness.
And what's fascinating here is the sheer density of the computational components suspended in that medium. I mean, we are talking about 80 to 100 billion neurons.
Billion, with a B.
Right. But they aren't functioning in isolation. They are supported by an equally massive, if not larger, infrastructure of glial cells.
Which are what exactly?
Well, these are your astrocytes, oligodendrocytes, microglia.
And for a long time, the scientific consensus treated glia as basically just biological mortar, right? Like just the glue holding the neurons in place.
Yeah, that was the historical view, absolutely. But modern neuroscience has completely overturned that. We now know glial cells are vital, active participants in the network.
Well, yeah.
They regulate the chemical environment, they provide structural scaffolding during that explosive fetal development you mentioned, and they actively speed up the electrical signals that neurons send.
Oh, that makes sense. Astrocytes even envelop synapses and can modulate how effectively two neurons communicate. So it's a highly dynamic, packed house.
But it isn't just a random homogeneous soup of 100 billion cells. It has a highly specific structural architecture built in distinct regions. Now, the sources bring up an evolutionary perspective here.
Yes, the historical context is really important.
They mention the classic triune brain model, the idea that we have this proto-reptilian core for survival wrapped in a paleo-mammalian limbic system for emotion, all topped off with the modern human neocortex for logic.
And I'm glad you brought that up because this raises an important question about how we view evolutionary neurobiology. The triune model is incredibly popular in pop psychology because, well, it makes for a great narrative.
Sure, sounds clean.
Right. But modern neuroscience shows that evolution doesn't just bolt new hardware onto old systems like a contractor adding a third floor to a house.
Right, it's not an archaeological dig where the bottom layer is perfectly preserved from some lizard ancestor. Exactly. Evolution is conservative, yes, but it integrates and modifies the entire stack. Having said that, there is a functional hierarchy that echoes our evolutionary lineage.
Okay, so how is it laid out then?
Well, at the base, connecting to the spinal cord, you do have the hindbrain and the brainstem structures, like the medulla and the pons, and these do handle the fundamental, non-negotiable homeostatic functions. Breathing, heart rate, blood pressure, the vomiting reflex.
Basically the autonomic autopilot.
Yes, exactly. You don't have to consciously remember to beat your heart, because that lower-level circuitry runs it flawlessly.
Thank goodness for that.
Seriously. Then, situated deeper in the central brain, you have the limbic structures. These developed roughly 250 million years after the hindbrain.
And this includes the hippocampi and the amygdala.
Right. This circuitry is deeply tied to emotional valency, the fight-or-flight response, and laying down new memories. It basically takes raw sensory data and survival instincts, and it assigns an emotional weight to them.
Got it. And then we get to the outer layer, the part that handles executive control, spatial reasoning, language, and conscious thought. The neocortex.
Which is massively prominent in humans. Interestingly, if you look at modern reptiles, their equivalent pallium or cortex is only 3 layers thick.
Oh, wow.
Yeah, but in mammals, and particularly primates, that architecture expanded into a highly complex 6-layered sheet of cells.
This highlights something that I found deeply compelling in the functional neuroanatomy notes. The brainstem and the limbic system are largely what we call nuclear structures.
Meaning what?
Meaning they are made up of nuclei, which are just dense, three-dimensional, balled-up clusters of neurons. But the neocortex relies on a cortical architecture. It's not a ball, it's a two-dimensional sheet.
Precisely. The computational neurons of the cortex are arranged in this thin layer, just a few millimeters thick, spread across the outer periphery. And the massive highways of axon fibers that connect them run underneath.
But this creates a massive engineering problem for biology. Because the neurons are organized in a two-dimensional sheet, they don't have to compete for 3D space the way a balled-up nucleus does.
Right, which allows for incredible complexity.
Exactly. But the human skull has a fixed, strictly limited volume. I mean, we have to pass through a human birth canal, so head size is capped. So how does evolution pack an ever-expanding 2D cortical sheet into a rigid, fixed-volume sphere?
Well, by introducing convolutions, the gyri, which are the ridges, and the sulci, which are the deep valleys.
Yeah, the sources have this great visual analogy for you. Imagine trying to fit a large cloth dinner napkin into a small water glass.
It's a perfect way to visualize it.
If you just lay it flat, the edges spill out. It won't fit. But if you wad it up, creating hundreds of complex folds, creases, and overlapping layers, you can stuff an absolutely massive surface area into a tiny confined space.
And that is exactly what the brain does. The more convoluted the brain, the more surface area it has, which scales directly with the species' capacity for complex computation.
That makes so much sense.
Yeah. And if we look closely at how this wadded up biological napkin is organized, we see it is divided lengthwise into 2 distinct cerebral hemispheres.
The left and the right, connected by the corpus callosum.
Yes. And the corpus callosum isn't just a bridge, it's a massive superhighway of roughly 200 million myelinated nerve fibers that allows the left hemisphere to synchronize constantly with the right.
200 million fighters just for communication. That's wild.
It is, and within these hemispheres, we have a clear physiological division of tissue. The outer layer, that wrinkled sheet, is the gray matter.
And it's gray because it is densely packed with the actual neuronal cell bodies and their dendrites, right? Like the local processor.
Exactly. And underneath that gray matter is the white matter. The white matter is essentially the long-distance cabling.
Why is it white, though?
It is white because the axons are coated in a fatty lipid substance called myelin, which is actively produced by those oligodendrocytes we mentioned earlier. Myelin acts like the rubber insulation on a copper wire. It prevents signal leakage and allows electrical impulses to travel incredibly fast over vast biological distances.
Now, within that outer bark of gray matter, the real estate is not homogeneous. The functional lectures talk about this concept of the eloquent cortex.
Oh, this is a crucial high-stakes concept, particularly in functional neurosurgery.
Right.
The eloquent cortex refers to specific, highly localized areas of the brain that directly control indispensable functions. If you suffer even minor damage to these precise areas from a stroke, a lesion, or surgical trauma, it produces a major, undeniable focal neurological deficit.
We're talking about areas like the primary motor cortex, which executes voluntary muscle movement, the primary somatosensory cortex, which maps touch and pain from your body, the primary visual and auditory cortices,
And perhaps most uniquely human, the distinct language centers.
like Broca's area.
Yes, Broca's area, typically localized in the left frontal lobe, which coordinates the intensely complex physical production of speech, and Wernicke's area, further back in the left temporal lobe, which handles the semantic comprehension of language.
So if we step back, we have this heavily folded, multi-layered hardware. A watery mass of gray and white matter highly organized into functional zones. But that leads us to a massive question.
Which is?
How do the individual components, these 100 billion neurons, actually run? What is the mechanism that powers this wetware?
To answer that, we have to look at the energy economics of the brain, which are frankly astonishing.
Yeah, the brain is astonishingly greedy. As we noted, it represents only about 2% of an average human's total body weight, but it commands 15% of your total cardiac output.
Exactly. Every time your heart beats, a huge surge of that blood volume is routed straight up your carotid arteries.
And it continuously consumes 20% of your body's total energy supply while at absolute rest.
It runs primarily on glucose, though it is highly adaptable and can run effectively on ketones during prolonged fasting or low carbohydrate states. But regardless of the fuel source, 1/5 of everything you consume goes exclusively to powering this 3-pound blob.
But here is the detail from the sources that truly shatters how we usually think about brain activity.
Oh, the 5% rule.
Yes, when you transition from just lying on the couch, staring blankly at a wall, to doing a highly complex, demanding cognitive task, like trying to solve a multivariable calculus equation or memorizing a new language, the brain's total energy consumption only goes up by about 5%.
It is a profound realization. What that tiny 5% delta tells us is that the resting brain is never actually resting.
Wow.
Yeah, the vast majority of the brain's massive energy budget is dedicated to intrinsic background operations. This is sometimes referred to as the dark energy of the brain.
Dark energy, I love that term.
It fits perfectly. It is continuously maintaining the steep electrical gradients of billions of cell membranes, actively pruning and strengthening connections, processing baseline sensory data, and crucially, generating complex predictions about the environment.
Okay, let's look at how that energy is physically used at the micro level to transfer information. Because it isn't like a continuous copper wire. How do two individual neurons actually talk to each other?
Well, neurons are highly polarized directional cells. They have distinct anatomical regions for receiving and sending.
Okay, lay it out for us.
On one end, you have the dendrites, which branch out like a complex, thorny tree canopy. These act as the receivers, collecting thousands of incoming signals.
Right.
Those signals filter down into the soma, the main cell body. If the cumulative signal is strong enough, the soma fires an electrical impulse down the axon, which is a single, long, thin cable that acts as the transmitter.
And that axon stretches out, sometimes over a meter long, to communicate with the dendrites of the next neuron in the chain. But here's the mind-bending physical reality they do not touch.
They absolutely do not.
Wait, you have 100 billion cells, forming hundreds of trillions of connections, creating a network orders of magnitude more complex than the global internet, and the cables aren't physically plugged into each other.
No, there is a physical, fluid-filled gap between the axon terminal of a sending neuron and the dendrite of the receiving neuron.
That is just so weird.
It is. That gap is the synaptic cleft. Now, on a macroscopic scale, it is incredibly small, only about 20 to 50 nanometers wide. But from the perspective of an electrical signal, it is an insurmountable chasm.
So it can't just jump across.
No, an electrical action potential traveling down the axon cannot just spark across that physical fluid gap.
So how does the information get across?
It has to undergo a radical phase change. It translates from an electrical signal into a chemical one.
Okay.
When the electrical action potential finally reaches the very end of the axon terminal, the sudden change in voltage forces specialized calcium channels to open. Calcium ions from the surrounding fluid rush into the cell.
And what does the calcium do?
This sudden calcium influx acts as a mechanical trigger. It causes tiny spherical storage vesicles packed with chemical messengers to fuse with the outer cell membrane.
And they spill their chemical contents out into the fluid gap.
Precisely. This biological mechanism is called exocytosis.
And those chemical messengers are the famous neurotransmitters we always hear about.
Right. They diffuse across that 20 to 50 nanometer gap and interact with highly specific receptor proteins embedded in the membrane of the postsynaptic neuron, the receiving cell's dendrite.
Now, you often hear this described as a lock and key mechanism. But based on the biochemistry notes, it's actually much more dynamic than that, right?
Oh, absolutely. The lock and key analogy is a bit too rigid. It's much more like a complex molecular handshake.
A handshake.
Yeah. When the specific neurotransmitter binds to the receptor, it doesn't just sit there. It causes the entire receptor protein to physically contort and change its three-dimensional shape. That physical contortion rips open a pore in the membrane, allowing charged ions to flood into or out of the receiving cell.
Wow. And the sources detail several of the big players that initiate this handshake.
Yes, the classic neurotransmitters.
You have glutamate, which is the brain's most abundant excitatory neurotransmitter. It's the primary chemical that screams, go to the next cell. Then you have GABA, the primary inhibitory transmitter, which essentially acts as the brakes, telling the next cell to quiet down.
And then overlaid on top of those fast-acting signals are the neuromodulators, which adjust the overall tone and sensitivity of large circuits.
Like dopamine, right?
Yes, dopamine, which is deeply involved in predicting reward, driving motivation, and regulating voluntary movement. And serotonin, which modulates overall mood, anxiety, and feelings of well-being.
So let's track the sequence here. Glutamate diffuses across the gap, initiates a molecular handshake, the receptor contorts, an ion channel opens.
Yep.
What happens next? How does a squirt of chemicals turn back into an electrical thought?
Okay, so when that channel opens, positively charged ions, usually sodium, rush into the receiving cell. This changes the electrical voltage of the local membrane. Because it was an excitatory transmitter like glutamate, it creates an excitatory postsynaptic potential, or EPSP. The internal voltage becomes more positive, moving closer to a critical firing threshold. It's a localized go signal.
And conversely, if it was GABA that bound to the receptor, it might open channels that let negatively charged chloride ions in. That creates an inhibitory postsynaptic potential, an IPSP.
Exactly.
The internal voltage drops further away from the firing threshold, acting as a stop signal.
Exactly. The chemical phase is over, and the signal has been successfully translated back into an electrical voltage change in the new cell.
Okay, let's unpack this part now. We've gone from the macro hardware to the micro power supply, and now we really need to look at the software. Because if neurons are just endlessly spitting go and stop chemicals at each other across microscopic gaps, how does that manifest as a complex cognition?
That is the million-dollar question.
Right. Like, how do you get from a microscopic squirt of glutamate to me recognizing the subtle sarcasm in a friend's voice?
Well, it all comes down to aggregation, math, and probability. At its most fundamental, irreducible level, a single biological neuron is essentially a microscopic adding machine.
An adding machine. Explain the math there.
Sure. A single neuron in the cortex might receive incoming synaptic contacts from up to 10,000 other distinct neurons.
10,000.
Yes. And at any given millisecond, hundreds of those upstream connections might be active simultaneously. Some are releasing glutamate, creating those positive EPSPs, telling it to fire. Others are releasing GABA, creating negative IPSPs, telling it to stay totally silent.
So the receiving neuron is constantly doing real-time, high-speed arithmetic.
Yes, a process we call synaptic integration. It continuously sums up these minute voltage changes, and it does this in two distinct ways. First, it sums them up in time-temporal summation. If one single synapse fires rapidly over and over, those sequential voltage bumps build upon each other.
Okay, that makes sense.
Second, it sums them up across its physical geometry spatial summation. It combines the signals arriving at completely different branches of its dendrites at the exact same moment.
So it's aggregating all the go signals and all the stop signals. And if the total mathematical sum of the EPSTs overpowers the IPSPs and pushes the cell's internal core voltage past a very specific threshold.
Boom! The neuron generates its own massive action potential, fires an electrical spike down its axon, and passes its own chemical signal along to the next layer of the network.
And if it doesn't reach the threshold?
If the aggregate sum doesn't reach the threshold, the neuron remains absolutely silent. Right. The inhibition successfully gated the excitation.
Okay, so that's the math of one single adding machine. But you hook billions of them together, and you get what the sources call micronetwork motifs.
Yes, motif.
These are the recurring basic circuit diagrams of the brain.
Yeah, as chaotic as the brain looks, we see the same highly conserved circuit patterns repeated over and over across different regions. For instance, you have feed-forward excitation. This is the simplest one. Neuron A excites neuron B, which excites neuron C, just passing a packet of information down a linear chain.
But if it was just feed-forward excitation everywhere, wouldn't the brain just instantly cascade into a massive epileptic seizure? Like, every thought would trigger every other thought.
Which is exactly why you have feed-forward inhibition.
Oh, okay.
In this motif, neuron A excites an inhibitory interneuron, which then releases GABA to shut down the next cell in the chain. It is an elegant mechanism to precisely limit the duration of a signal or to sharpen the contrast of a sensory input by silencing the neighboring neurons.
The functional notes also emphasize convergence and divergence.
Right. Convergence is basically a data compression mechanism. This is when hundreds or thousands of different neurons all send their axons to synapse onto one single target cell.
Okay, so they all funnel in.
Exactly. That single cell is gathering highly diverse, disparate information from across a wide network and synthesizing it into a single output.
And divergence is the opposite.
Divergence is the exact opposite. One single neuron branches its axon out to contact thousands of different target cells, allowing it to broadcast a vital message incredibly widely.
And then there is recurrent excitation. And from reading the texts, this seems absolutely paramount for things like memory and sustained attention.
It is fundamental.
Like neuron A fires and excites neuron B, but neuron B has a collateral branch that loops backward and excites neuron A again while also exciting its neighbors. It creates a self-sustaining, reverberating loop of electrical activity.
Those recurrent circuits are the architectural foundation of what computational neuroscientists call auto-association networks. We will actually explore them deeply when we discuss how the brain physically writes memories.
Here's where it gets really interesting, though, because everything we just talked about, the adding machine, the recurrent circuits, the spatial summation, that's just the mechanical signaling.
Right, it's the physics.
It doesn't explain what the information actually means or how a coherent subjective reality emerges from a bunch of ions moving across membrane.
No, it doesn't.
For that, the sources pivot to a wildly complex, fiercely debated theory unifying physics, neuroscience, and AI, known as the free energy principle, and its behavioral counterpart, active inference.
This is a monumental theoretical shift. Pioneered largely by Carl Friston, this framework attempts to provide a single, unified mathematical equation for all brain function and, frankly, all biological self-organization.
That's a huge claim.
It is. The foundational premise is rooted in thermodynamics. Any adaptive living agent must maintain its physical and biological boundaries to survive. It cannot succumb to the second law of thermodynamics. It cannot just dissolve into the chaotic entropy of its environment.
Right, like a fish needs to stay in the water. If it wanders onto land, it loses biological boundaries and dies. So, to maintain those boundaries, the brain has to minimize the informational entropy of its sensory inputs.
In plain English, the brain is fundamentally driven to minimize mathematical surprises. Yeah. Now, we aren't talking about the psychological emotion of being surprised at a surprise birthday party. We mean statistical surprise. The brain wants its incoming sensory data to consistently fall within a tight, predictable, biologically safe range.
If we connect this to the bigger picture, think about the physical reality of the brain. The brain is locked inside a dark, silent, thick bone vault. It has absolutely zero direct access to the outside world.
None.
It doesn't see light. It doesn't hear sound. All it receives is a continuous, chaotic barrage of raw electrical spikes from the optic nerves, the auditory nerves, the sensory receptors in the skin. To survive, the brain has to constantly guess what external physical phenomena are causing those specific patterns of electrical spikes.
So how does it do that? It builds a model. Under active inference, the brain constructs a deeply complex generative model of the physical dynamics of the world, alongside a recognition model of its own internal sensory states. Because it is trapped in the dark, it operates by constantly generating top-down predictions about what it expects to feel, see, and hear in the next millisecond based on past experience.
Wait, so it doesn't just passively wait for data. It predicts the data before it arrives.
Exactly. It then continuously compares those top-down predictions against the actual bottom-up sensory data arriving from the peripheral organs. experience.
And if there is a mismatch between what the brain predicted and what the sensory organs actually report, that mismatch is mathematically defined as free energy, or simply a prediction error.
Yes.
And according to this framework, the brain's singular ultimate objective is to continuously reduce that discrepancy.
Now, there are two distinct, equally valid ways the biological system can reduce that prediction error. And this is the absolute genius of the active inference framework.
I really love this part of the reading. Under active inference, beliefs, which are your internal predictions about what you are currently sensing, and intentions, which are your desires to physically move your body, are treated mathematically as the exact same phenomenon.
They are.
The text uses a great unified term for this: expectance. A belief and a motor command are both just statistical conceptions of the immediate future. The only difference is how the brain chooses to resolve a violation of that expectance.
Precisely. Let's ground this in an example.
Perfect.
Say your brain's generative model is predicting that you are currently standing in a warm, comfortable room. But your peripheral skin sensors suddenly send up a barrage of spikes indicating freezing cold.
Yikes.
You now have a massive prediction error. Free energy is high. Option 1 to resolve it, you change your internal model to match the incoming reality. You update your belief. The brain accepts, okay, my previous model was wrong. I am actually in a freezing room. That updating process is what we call perception.
So you change your mind to fit the world.
But there is option 2. You refuse to update the model. You keep your prediction of warmth firmly in place as an active goal, and you physically change the external reality to force the sensory data to match your stubborn model. You execute a motor plan to walk over and turn up the thermostat, or you put on a heavy sweater until your skin sensors finally report the exact warmth your brain was stubbornly predicting. That process is called action.
You change the world to fit your mind.
Exactly. Active inference completely dissolves the traditional boundary between sensory processing and motor control. It posits that perception and action are just two symmetric ways of solving the exact same mathematical problem.
That's brilliant.
Both exist solely to minimize the free energy, to crush the discrepancy between the brain's internal model and the external reality.
That is just incredible. It completely reframes human behavior. So, moving from the math of prediction into biological function, now that we understand the brain is this relentless prediction engine running on wet networks, how does it physically partition the work? Like, how does it carve up the processing of the reality around you?
To process that reality efficiently, the highly convoluted cerebral cortex is divided anatomically into large sections called lobes.
Right.
And while modern neuroscience has entirely moved away from the rigid phrenological, one area does strictly one thing, view networks are highly distributed. Certain lobes undeniably serve as the primary hubs for specific sensory and cognitive processing streams.
Let's break down the functional geography of those lobes. First, you have the occipital lobe, situated right at the back of the skull.
This is the territory of the primary visual cortex. When photons hit your retina, the cells translate that light into electrical spikes. That signal travels deep into the center of the brain to a crucial relay station called the thalamus, and then it is projected straight to the back of the head.
The back of the head, okay.
The occipital lobe is where visual input is first consciously processed, but at this stage, it is highly fragmented. It is just detecting the orientation of lines, contrasting edges, and flashes of light.
Right. Just because a cluster of neurons detects a jagged diagonal line doesn't mean you know you're looking at a wolf.
No, absolutely not. From the primary visual cortex in the occipital lobe, the visual information splits and is sent forward along two distinct association pathways to figure out what that line actually means.
Where do they go?
One pathway routes upward into the parietal lobe. This is known as the dorsal stream. The parietal lobe houses the somatosensory cortex, handling complex touch and pain, but it is also the brain's primary spatial processing center.
Okay.
The dorsal stream is often called the where pathway. It calculates precisely where an object is in 3D space, its trajectory, and how fast it is moving relative to your body.
And the second pathway goes downward into the temporal lobe.
The ventral stream. The temporal lobes sit on the sides of your head, roughly behind your ears. They are the primary centers for auditory processing, but the visual information routed down here is processed to determine exactly what an object is.
The what pathway?
Yes. This is where highly complex feature extraction occurs: facial recognition, object naming, reading text, and color interpretation. And crucially, the temporal lobe also houses the hippocampus deeper inside, which means this what stream is intimately functionally tied to cross-referencing objects with your past memories.
And then finally, sitting at the very front of the brain and taking up a disproportionately massive amount of volume in humans compared to other mammals is the frontal lobe.
The integration center, specifically the prefrontal cortex, or PFC.
What does the PFC do?
I like to think of it not so much as a CEO handing down rigid orders, but rather an orchestra conductor. It receives highly processed, fully formed data streams from all the other sensory lobes. It manages your working memory, sustains your attention, houses your personality, and critically exerts top-down inhibitory control over impulses.
Wow, it really does everything.
It evaluates the complete situation and plans the complex execution of future movement.
The sources provide this perfect, highly illustrative example that demonstrates how all these separate lobes in discrete networks fire synchronously together in a fraction of a second to keep you alive. The alligator example.
It's a classic sequence. Walk us through the neuroanatomy of it.
Okay, so imagine you're walking near a murky swamp in Florida, and you look down at the bank. Your eyes capture an image. That raw sensory data goes through the thalamus directly to the occipital lobe at the back of your head. The occipital lobe fires and essentially says, okay, I see a long, dark green shape with jagged edges and a textured surface.
And that raw feature data immediately splits. It travels up the dorsal stream to the parietal lobe, which rapidly calculates that distinct shape is exactly 6 feet away. It is currently stationary, but it is oriented directly toward our center of mass.
Simultaneously, the exact same data travels down the ventral stream to the temporal lobe, which works to identify the object. That is a biological entity, animal. It instantly pings the hippocampus, frantically digging through your stored auto-association networks. Have we seen this specific pattern of lines and colors before? Yes. That matches the template for an alligator. Alligators are apex predators.
And instantly, that identification triggers the amygdala in the limbic system, flooding your system with adrenaline and sounding a massive physiological alarm.
Adrenaline spike, boom!
Boom! Now, all of this highly processed data, the precise spatial location, the semantic identification, the intense emotional memory of danger is instantly routed forward to the prefrontal cortex in the frontal lobe.
The PFC acts as the final executive conductor. It takes all the incoming data, compares it against its internal generative model of survival, and makes a definitive judgment call. We do not want to be near an apex predator. We need to evade.
Exactly.
It immediately sends a complex motor plan to the premotor cortex to plot an escape trajectory, which then signals the primary motor cortex.
And the primary motor cortex fires a massive coordinated volley of electrical impulses down through your brainstem, down your spinal cord, and out to the massive muscle groups in your legs, causing you to violently jump backward.
All of that.
All of this computation, from the moment a photon bounces off the alligator's scale and hits your eye, to the massive muscle contraction in your calves, happens seamlessly in just a few hundred milliseconds. It is an astonishing symphony of network integration.
But it's not always just a cold, instantaneous calculation, is it? When the PFC decides what to do in less obvious situations, there is a fierce internal debate happening across the networks. How do we actually make decisions, especially when those decisions involve significant risk?
This brings us to a prominent framework in cognitive neuroscience known as the dual systems model of decision-making.
Okay, dual systems.
It posits that decision-making is essentially a constant neurological tug-of-war. On one side, you have the prefrontal cortex, the rational, deliberative, slow-thinking brain. It calculates long-term consequences, abstract probabilities, and moral implications.
And the other side?
But it is in a constant struggle for dominance with the limbic system, particularly the amygdala and the striatum, the fast, emotional, impulsive, deeply survival-oriented brain.
And the outcome of that tug-of-war isn't fixed, right? It changes drastically depending on the neurochemical environment at that exact moment.
Exactly. The balance of power is highly sensitive to neuromodulators. For instance, when dopamine is heavily present in the striatum, say, in anticipation of a massive salient reward, like a huge sum of money, high-calorie food, or a surge of social status, it artificially inflates the perceived value of the choice.
Wow, okay.
Simultaneously, it can suppress the PFC's natural risk aversion signals. The emotional reward-seeking brain effectively shouts louder than the rational, calculating brain, leading to highly impulsive risk-taking behavior.
Is that why teenagers do such crazy things?
Actually, yes. We see this neurobiological dynamic most pronounced during adolescent development. The subcortical, socio-emotional, and reward systems mature years faster than the prefrontal executive control systems, creating a temporary window of intense vulnerability to risky behavior.
Which perfectly sets up the next phase of our deep dive behavior. Once the frontal lobe finally makes the decision, whether it's to jump back from an alligator or take a calculated risk on a new venture. How does the brain actually orchestrate that physical interaction with the world? How do we translate thought into action?
We mentioned the primary motor cortex earlier, located at the very back of the frontal lobe. But what's truly startling for most people to learn is that roughly 80% of the complex motor circuitry in your brain operates completely beneath your conscious awareness.
Right, 80%. So when I consciously decide to reach out and pick up my coffee cup, I'm really only aware of a tiny fraction of the necessary processing.
Much less than a fraction, really. The prefrontal cortex makes the high-level, conscious, executive decision. I desire the coffee, pick up the cup. But the PFC has absolutely no idea how to execute that.
It doesn't.
No, it doesn't know the exact tension required in the bicep or the precise micro-adjustments of the radial wrist muscles. It just delegates the intention. The abstract command goes from the PFC to the primary motor cortex.
And the anatomical sources describe the primary motor cortex as being organized topographically, the famous motor homunculus map.
Yes. If you were to literally drape a map of the human body over the strip of the motor cortex, it's not proportional to your physical size at all. It is heavily, disproportionately biased toward anatomical areas that require incredibly fine, complex motor control.
So like the hands.
Yes, so on this neurological map, the representations for the face, the lips, the tongue, and the hands are absolutely massive, taking up huge swaths of tissue, while the representations for the large muscles of the torso and legs are relatively tiny.
Okay, so the motor cortex gets the abstract order from the PFC, but it still doesn't handle all the fine-tuning of the muscles by itself.
No, it needs massive computational support. From the motor cortex, the signal loops down deep into the basal ganglia, which acts like a library storing pre-learned automated motor programs and routes through the thalamus. But crucially, before the final signal is sent down the spine, it communicates heavily with the cerebellum.
The cerebellum. The deeply ridged little brain sitting tucked under the back of the skull.
Yes. It only accounts for about 11% of the brain's total volume. But due to its incredibly dense crystalline cellular architecture, it houses over half of the total neurons in the entire human brain.
Half the neurons.
Yes. For over a century, classical neurology taught that the cerebellum was purely a motor coordinator. It takes the intended motor command from the cortex, continuously compares it to the actual real-time proprioceptive feedback coming from the physical joints and muscles, and instantaneously smooths out the errors so you smoothly grasp the coffee cup instead of wildly punching it off the table.
But the updated research sources reveal something completely revolutionary about the cerebellum's true function.
This is one of the most exciting breakthroughs in modern neuroanatomy. Researchers recently utilized highly advanced viral tracer techniques.
Wait, viral tracer?
Yeah, they took a specially modified strain of the herpes virus because the herpes virus naturally travels along nerve fibers and injected it into the nervous system. Because they engineered this specific virus to jump backwards across the synaptic clefts in a strictly time-dependent manner, researchers could perfectly trace functional neural pathways 2, 3, or 4 synapses deep, mapping the exact network hierarchy.
And what did the viral tracers reveal?
They definitively proved that the cerebellum forms continuous closed-loop circuits with the cerebral cortex. Now, we already knew it formed these loops with the primary motor cortex, but the viral tracers proved it also forms distinct, massive, closed loops with purely non-motor areas, specifically Area 46 in the prefrontal cortex.
Area 46, the cognitive executive CEO area.
Exactly. The implications of this are profound. It strongly implies that the dense computational power of the cerebellum isn't just dedicated to smoothing out physical and muscular movements. It is actively smoothing out and refining purely cognitive and emotional processes using the exact same predictive computational architecture.
That is just profound.
It helps coordinate, sequence, and error correct our abstract thoughts and emotional regulations just as much as it coordinates our physical limbs.
It shows how deeply intertwined cognitive thought and physical action really are, which naturally brings us to a behavior even more complex than picking up a cup or jumping from an alligator.
Yeah.
Human speech. Why do we speak, and how does the brain actually coordinate it?
Generating spoken language is arguably the single most complex, highly orchestrated behavior the human brain produces. It doesn't rely on just one area. It relies on an intricate, highly synchronized network of at least 8 distinct major white matter pathways connecting widespread cortical regions.
We mentioned the 2 main hubs, Broca's and Wernicke's areas, earlier.
Yes. Let's trace the pathway of a spoken sentence. Wernicke's area in the temporal lobe is furiously working to comprehend the semantic meaning of the abstract concepts you want to convey. It translates thought into linguistic structure.
Okay, then what?
It then sends that dense packet of information forward, primarily via a massive fiber tract called the arcuate fasciculus, directly to Broca's area in the frontal lobe. Broca's area is the translator. It takes that abstract linguistic code and translates it into a highly sequenced, millisecond perfect physical motor plan.
Millisecond, perfect.
Just the seemingly simple act of speaking a single word requires the flawless coordination of roughly 35 different muscles across the face, the tongue, the larynx, and the diaphragm.
Now, I have to push back a little on the anatomy here. The functional texts say that we know generally where these language areas are localized, left frontal, left temporal.
Sure.
But they also explicitly state that the exact microtopography of language is highly variable between different individuals. So if I'm a neurosurgeon and I'm about to remove a tumor from a patient's left hemisphere, I can't just look at a standard anatomy textbook to know exactly where their specific Broca's area starts and stops.
You absolutely cannot. The visible anatomical landmarks, the specific folds of the gyri are not reliable enough when millimeter-level precision is required.
Wow.
The language networks can shift slightly based on individual development. If a surgeon cuts even 1 or 2 millimeters into the eloquent language cortex, the patient could wake up with permanent, devastating, expressive aphasia. They might never speak a coherent sentence again.
So how do they possibly avoid it when every brain is uniquely wired?
This functional variability necessitates the incredible, almost surreal procedure of awake cortical mapping. It is the absolute gold standard for resecting tumors located anywhere near eloquent areas.
I've heard of this.
The patient is placed under general anesthesia just long enough to open the scalp and the skull because the skin and bone are heavily innervated with pain receptors. But the actual tissue of the brain itself has absolutely 0 pain receptors. You cannot feel your own brain.
So they literally wake the patient up in the middle of the operation.
They do. While the brain is completely exposed in the operating theater, the patient is brought out of anesthesia and is fully awake and conversing.
That is just nuts.
The neurosurgeon uses a specialized probe to apply tiny, precise electrical currents directly to the surface of the cortex. This mild current temporarily stuns that specific millimeter of tissue, briefly taking it offline. Meanwhile, a clinical neuropsychologist is sitting face-to-face with the patient, having them continuously count out loud or name pictures on a flashcard. If the surgeon stimulates a specific spot and the patient suddenly stops speaking mid-sentence, or their words suddenly slur into gibberish,
The surgeon knows immediately, okay, do not cut there. That specific millimeter of tissue is a critical node in their unique language network.
Exactly. They meticulously map the individual's unique functional neuroanatomy in real time, placing tiny sterile numbered tags directly on the brain surface. This allows them to maximize the removal of the tumor while perfectly preserving the highly idiosyncratic, eloquent pathways.
It is absolute science fiction that this is a routine procedure happening in hospitals every single day. So we've covered the hardware, the software, the computational theories, and how the brain dynamically controls our behavior. But here is the final, perhaps most important piece of the puzzle adaptation.
The plastic brain.
Exactly. The brain isn't a static machine that just degrades over time. The very act of behaving, of experiencing the world, physically and chemically, rewires the hardware.
This brings us to the profound concept of neuroplasticity. The brain is remarkably inherently malleable. It is most aggressive during early childhood development, but this structural plasticity continues every single day throughout your entire life.
How does that actually work mechanically? How do we learn? Like, how are memories formed at the physical, cellular level?
The foundational principle of neuroplasticity is often summarized by the famous axiom known as Hebb's rule nons that fire together, wire together.
Which means what, exactly, in terms of chemistry?
It refers to a mechanism called activity-dependent plasticity. When a presynaptic neuron repeatedly, persistently, and successfully activates a postsynaptic neuron, a cascade of complex biochemical changes occurs in one or both cells that physically alters their anatomy, making that specific connection stronger, faster, and more metabolically efficient.
The molecular biology sources mention the critical role of neurotrophins here.
Yes, specifically complex protein molecules like BDNF, brain-derived neurotrophic factor. Think of BDNF as high-powered biological fertilizer.
Okay.
When a postsynaptic cell is strongly and repeatedly activated, it synthesizes and releases BDNF backward across the synaptic cleft. This chemical binds to the presynaptic axon. It promotes cellular survival, actively prevents programmed cell death, and actually causes the axon to sprout new branches and the synapse to grow physically larger.
And this ties perfectly back to those recurrent excitatory circuits we discussed earlier, particularly in the hippocampus.
Exactly, the autoassociation networks. Because the neurons in the hippocampus are wired to loop back and continuously stimulate each other, a highly specific pattern of sensory input, say the visual shape of your grandmother's face, combined with the sound of her voice, will cause a specific widespread web of neurons to fire simultaneously.
And because they fire together?
Because they are firing together in a sustained loop, they wire together. The thousands of synapses connecting that specific web all get flooded with BDNF and physically strengthen.
So the memory isn't just stored inside one single isolated grandmother cell.
No, the memory is distributed across the entire architecture of the network. The highly specific pattern of synaptic strength is the memory. Years later, if you only receive a partial sensory cue, like just smelling the specific brand of perfume your grandmother wore, it strongly activates just a small fraction of that original web.
Wow.
But because the internal connections were strengthened by BDNF, the electrical activation rapidly cascades and spreads through the entire network, and you instantly recall the complete multisensory memory associated with that smell.
That elegantly explains how we learn internally, how we lay down associative memories. But how does sustained physical practice and interaction with the environment actually change the macroscopic structure of the brain?
It's amazing to see at the macro level.
The sources highlight two deeply fascinating illustrative examples of this. Let's start with the classic animal studies, the kittens.
This is a foundational, though by modern standards somewhat controversial, study in sensory neurodevelopment. Researchers took newborn kittens and raised them during their critical window of visual development in a highly controlled environment where they only ever saw vertical lines.
Just vertical lines.
The inside of their enclosure was painted with vertical stripes. They never saw a horizontal line, no diagonals, no complex shapes, just verticals.
And what was the neurological result of that restricted environment?
When the cats reached maturity, they were removed and placed in a normal, highly complex visual environment. The researchers found that the cats were completely, literally blind to horizontal lines. They could see chair legs perfectly and navigate around them, but they would walk face first right into a horizontal bar suspended in front of them.
Why? Did the restricted environment damage their retinas? Did their eyes stop working?
No, their eyes and optic nerves functioned perfectly. The deficit was entirely cortical. In the primary visual cortex of a normal brain, there are distinct columns of neurons dedicated to processing lines of every possible orientation. But in these kittens, the specific neurons that naturally would have processed horizontal lines had never been stimulated.
Oh, wow.
Because they never fired, they never triggered the release of BDNF. They were systematically outcompeted for synaptic real estate by the hyperactive neurons processing vertical lines. The unused horizontal connections literally withered, retracted, and died off.
It is use it or lose it, enacted at a brutal biological level.
Precisely. The brain aggressively adapted its physical hardware to perfectly match the statistical reality of the limited environment it was raised in. It optimized for verticality because, as far as the brain's generative model knew, horizontal lines simply did not exist in the universe.
The second example from the text is much more uplifting, dealing with human expertise, the musician study.
Yes. Researchers used advanced neuroimaging to look at the brains of world-class professional string musicians, specifically violinists and cellists, who began their intensive physical training in early childhood. They specifically examined the parietal lobe, looking at that somatosensory homunculus map we discussed, the map of physical touch on the body.
Right.
For a right-handed violinist, the fingers of the left hand are constantly engaged in incredibly complex, rapid, independent movements on the strings, requiring immense continuous sensory feedback. The fingers of the right hand, conversely, simply grip and draw the bow, a much grosser motor movement.
Okay, so big imbalance there.
When researchers mapped their cortices, they found that the physical cortical representation, the actual volume of brain tissue dedicated exclusively to the left-hand fingers, was massively expanded compared to their own right hand, and massively expanded compared to non-musicians.
So by relentlessly practicing scales and concertos for tens of thousands of hours, they literally commanded their brain to grow more dendritic branches and dynamically reallocate more physical tissue to those specific fingertips. The software forced the hardware to structurally upgrade.
That is exactly the mechanism at play. The repeated behavioral demand drove structural hypertrophy in the relevant circuits.
But there is a dark side to this neurodevelopment, right?
Yeah.
We've talked extensively about synapses strengthening, dendrites growing, and maps expanding. But a massive, arguably more important part of brain development is actually about targeted destruction.
Yes. The biological process of apoptosis, which is genetically programmed cell death, alongside massive synaptic pruning.
The developmental sources point out a staggering statistic. The fetal brain vastly overproduces neurons, sometimes generating up to 50% more cells that will actually survive into adulthood.
50% more.
Why would biology expend massive amounts of energy to build millions of complex cells just to intentionally kill them?
Because it is a highly effective, albeit brutal, evolutionary strategy for ensuring incredibly precise, custom-tailored wiring. The genetic code isn't large enough to blueprint every single one of the trillions of synaptic connections. So, the developing brain throws out a massive excess of neurons and initial connections, and then forces them to compete for limited resources like BDNF or physical space on target dendrites.
Survival of the fittest inside your head.
Exactly. The connections that successfully carry meaningful, synchronized information survive and get structurally reinforced.
And the losers of that biological competition.
The neurons whose axons arrive too late to the target, or the ones that fire randomly out of synchrony with the rest of the network, fail to secure enough neurotropic support. The lack of BDNF activates their own internal suicide genes. The cells literally dismantle their own DNA, self-destruct, and are quietly absorbed as cellular debris by the surrounding microglia.
That is wild.
The scale of this destruction is staggering. In the infant brain, during the peak refinement of the corpus callosum, that massive bridge between the hemispheres, the brain actively eliminates up to 60 axons every single second.
60 microscopic cables actively severed every second, just to prune away the noise and refine the signal, which perfectly contextualizes this final fascinating study from the sources on how human intelligence actually develops.
Yes, the comparative fMRI studies looking at the neurobiology of executive functioning in young children versus mature adults.
Researchers put kids and adults into fMRI scanners and had to perform the exact same complex cognitive tasks, things requiring deep focus and impulse control. And they found that early in life, when a child attends an executive function task, the metabolic activity in their prefrontal cortex is weak, but is wildly widespread.
It's highly diffuse.
Right, a massive region of the frontal lobe is lighting up, but dimly.
But as those children grow, learn, practice, and master these cognitive tasks, the fMRI activation pattern changes entirely. When healthy adults perform the exact same task, the activity becomes highly robust, incredibly intense, but strictly focal. It's a brilliant, tightly concentrated spot of metabolic activity.
So, synthesizing everything we've covered today, what does this all mean?
It means that cognitive development, mastery, and intelligence aren't just about growing a physically bigger brain or mindlessly adding more and more connections. It is fundamentally about pruning away the noise. In the child's highly unpruned brain, the diffuse activity indicates that the region has to work incredibly hard, loudly expending a massive amount of metabolic energy across an unrefined, noisy network, just to achieve a basic baseline level of cognitive performance.
But through years of education, practice, environmental interaction, and that brutal, relentless apoptosis and synaptic pruning, all the inefficient, noisy connections are systematically stripped away.
Exactly. The remaining, highly refined circuits function with absolute maximum energy efficiency. That robust, tightly focal fMRI activity in the adult brain represents a highly optimized, streamlined neural network capable of supporting incredibly advanced executive function with an absolute minimum of wasted metabolic energy.
And that brings us perfectly full circle on our deep dive. We started with a staggering 250,000 cells being born every minute in the womb, generating a massive, chaotic overproduction of watery, fatty hardware.
Billions of microscopic adding machines separated by tiny fluid gaps, communicating via molecular handshakes and organizing into massive recurrent loops.
And through the relentless, brutal competition of experiencing the world, the brain aggressively prunes itself into a sleek, metabolically highly efficient prediction engine.
An engine solely designed to minimize mathematical surprise and keep your biological boundaries intact in a wildly unpredictable universe.
Which, if we really sit with it, leaves us with a rather profound, perhaps slightly unsettling thought to close on, drawing directly from the implications of Carl Friston's active inference framework.
Go for it. Leave us with something to think about.
We established very clearly that your physical brain is strictly locked inside a dark, silent sensory deprivation vault made of bone.
Completely isolated.
It has absolutely zero direct, unmediated contact with the outside world. It merely receives a constant chaotic stream of raw electrical spikes from the peripheral nerves. And in response, it furiously generates top-down predictive models to make sense of that noise, actively minimizing its free energy, desperately trying to guess what is out there causing the spikes.
Right, the brain's best guess.
If that mathematical framework is true, is the subjective reality you're experiencing at this exact moment, the room you're sitting in, the vivid colors you think you see, the specific pitch and tone of our voices, is any of it actually the objective real world? Or is everything you have ever known, felt, or loved simply your brain's most highly optimized, metabolically efficient hallucination?
Wow. Keep questioning your reality, everyone, and never stop exploring the deep dive of